All right, good morning. Welcome to our first seminar for the semester. Today I'm going to introduce you to Stephen O'Keefe. He's been a professor of medicine at the University of Pittsburgh Medical School since 2003. He graduated in medicine at Guy's Hospital in London in 1972 and received his MD by thesis in 1980. In addition, he studied and was awarded for a Master of Science in Human Nutrition. I'm sorry, I just ran here. So happens when they oversell the parking spaces in Lot 100. Okay. Sorry. He was awarded a Master of Science in Human Nutrition at the London School of Hygiene and Tropical Medicine in 1974. He trained in internal medicine at Oxford and was a fellow in internal medicine at Harvard and MIT. His interest is in the research and practice of nutritional gastroenterology, which took him to South Africa and then to the Mayo Clinic. He's had a lifelong interest in geographical differences in disease patterns, and currently is examining microbiota, metabolomic mechanisms. Oh my goodness, I'm sorry. I'm so sorry. With NIH support that explains why the chief determinant of colon cancer is diet, and why African Americans have the highest risk of colon cancer in the USA, while rural Africans rarely get the disease. Today he will speak to us on the topic of the critical role of the diet in colon cancer. So please, with that crazy introduction, help me welcome Dr. Stephen O'Keefe.
Thank you. That's the nicest introduction I ever had. So today I'll be sharing with you my prejudices about this particular topic, which is all on our minds because we're all interested in what we eat and how it affects our health. So just to start off, this doesn't advance.
Sorry, I didn't hear them.
I can manually do it if you want.
Oh, no. There we go.
There we go. All right. Okay. It's electronic age. So what's interesting about colon cancer is that it varies 100-fold around the world. And if you look at the geographical differences, you can see that Africa and that sort of area along the equator here is a very, very low incidence area, whereas high incidence areas are basically across westernized countries. And the studies, epidemiological studies done from all these different parts of the world have looked at the relationship between cancer incidence and what you eat. And they've identified certain dietary components such as red meat, animal fat, processed meats, also other factors in lifestyle such as obesity and inactivity, too much alcohol and cigarettes, which increase risk, and other factors in the diet, particularly fiber, which is associated with a reduction in risk, vegetables, particularly green and leafy vegetables, calcium, and then other dietary factors such as fish oils, which you guys are particularly interested in, antioxidants, selenium, folic acid, and also hormones can affect risk as well. So, what's interesting about this is that there's a group of world-renowned epidemiologists at was in Oxford, England, called Doll and Pito, and using this information, they were able to estimate that over 90% of all GI cancers were diet-related. The great thing about that is the other side of it is that if that really is true, over 90% are actually preventable by dietary change. Whoops, this one here. So let's just start off with a few notes about these factors that I've talked about. Fiber is basically a complex carbohydrate that is not digested and ends up in the colon and is therefore food for the microbiota. Resistant starch previously was considered a digestible starch, but it isn't, and so therefore it's not considered one of the fibers as well. So there's an expanding range of different types of fiber. And the protective effect of fiber was first noted by Burkitt in his studies before the war, which showed that African traditional diet had a high fiber content and colonic cancer was virtually unheard of. There was a certain amount of controversy regarding relationship between fiber and cancer risk produced by Giovanetti and people in America from Tufts. But a better study was actually published by the European group EPIC, which concluded that, basically they looked at a whole sway of different countries, and they all had various different fiber intakes from high to low, and so they were better to examine the spectrum, and they found that a doubling of the fiber intake was associated with a 40% reduction in colonic cancer incidence. So, there's no doubt that, if you go from a low to a high fiber content of your diet, it has a major protective role. Animal meats and fat, again, the World Cancer Association has shown that fiber, red meat, are extremely well associated with cancer risk, and fat to a more moderate degree, and fruit and vegetables also to a more moderate degree. There are a number of different factors in meat, for instance, which have been shown experimentally to be carcinogenic, such as the formation of heterocyclic amines if you overcook meat, browning, so to speak. Also the presence of heme, which actually injures the colonic epithelium and increases proliferation. And then with regard to fat, there are also multiple mechanisms whereby it might affect colon cancer risk. Again, mainly by its effect. If you eat a high-fat diet, you stimulate hepatic bile acid synthesis. They go through, a proportion escape, go into the colon, and then metabolized by bacteria to secondary bile acids, which are strongly carcinogenic. Some free fatty acids themselves have also been shown to produce proliferation in the colon, so that's a more direct method. And then there's this fascinating study of Proliferation. So if you eat a high-fat saturated diet, you increase the synthesis of taurocholate, which is one of the fatty acids, has a high sulfur content. It goes into the colon, it stimulates Proliferation to produce hydrogen sulfide, which is genotoxic and pro-carcinogenic. So there are multiple mechanisms associated with the food effect. The association between fish consumption, one's often felt that people who eat a lot of fish should actually be protected as well because of their omega-3 fatty acid concentrations or content. This is a meta-analysis that was done, which is really all over the place, and is probably swayed by this study here towards showing some favor towards, a high fish diet suppressing colon cancer risk. But I don't know if any of you have read this, but there's so much replication between studies here. They split off males and females and report the same study twice. So, students always beware of meta-analyses because they don't really mean very much. You need one good solid study to give you a proper answer to this. So we're not sure about that. Fruit and vegetables, again, it's difficult to know what specific role they have because generally if you eat healthy, you eat a lot of fruit and vegetables. But they do contain things such as vitamins. Whoops. Where have we gone? I went through very fast. Vitamin C, antioxidants, folic acid, selenium, calcium, which all suppress cancer risk epidemiologically and in experimental models. And then they contain bioactive compounds within cell walls, such as phenolics, which again are anti-carcinogenic. One of the most fascinating studies was this on Japanese migrants to Hawaii. And at the turn of the 19th, 20th century, there was an influx of Japanese to Hawaii to work on sugar farms, I think it was. And at that time, colon cancer was almost unheard of in Japan. And within one generation of moving over to Hawaii, they actually achieved basically the same incidence rate as native Hawaiians, showing that it only needs one generation of change to westernized country before you change your rate of colon cancer. And they examined the diet and they correlated it with a high red meat, low fiber intake diet. So westernized colon cancer is one of the westernized diseases. And the westernization has been associated with dramatic increases in expected lifespan from less than 25 years before the Industrial Revolution to over 80 today. So we've obviously made greater advances. This change, however, is too rapidly to be attributed to genetic evolution. That takes thousands and thousands of years. But it can be to the remarkable ability of humans to adapt to their environment, to avoid life-threatening events such as perinatal complications, acute infections, trauma, and war, except I don't know how good we are at avoiding war at the moment. And basically what's happened is that it's exposed the people who survived through this to this age with the emergence of westernized diseases, which, as I say, includes colon cancer, but it's heart disease, pancreatic breast cancer as well. And it's been shown now that these westernized diseases are considered the most serious threat to public health in the USA today. So there's a desperate need to be able to identify what's wrong and how we can fix it. So what about food? From what we know of the process of evolution, the dietary needs of every organism are genetically determined. And some have argued that our health will be better maintained by the diet first established by Homo sapiens in the Paleolithic era in Africa, a period that lasted from 2.5 million years ago to 11,000 years ago. So that is sort of diet that our genes were basically developed with. And if we look at coprolite findings from people from the remnants of those populations, the analysis has actually indicated that they had a high carbohydrate diet. So what is wrong with the westernized diet? And basically, you know, there's too much grilled and fried foods. There's too much fat. There's too much carbohydrate, simple carbohydrate, not resistant carbohydrate. Alcohol comes into the equation if you drink too much. Preservatives and ketchup and various other things that make food so delicious. And you'll note the deficiency of fruit and vegetables, and particularly in fiber. So, you know, what we're left with is something which is sumptuous. It's absolutely delicious. We all love to have a good burger and fries, particularly at a football game. But, you know, it's not particularly what we were designed to eat. So what I'm going to show in my talk today is that, you know, it's not actually diet that determines cancer risk, but it's the effect diet has on the microbiome to produce things that are either good or bad for the colon. So if we start off with a little graphic, we have food coming into an idealized GI tract. This is the small intestine, and this is the large over here. And most of our food, about 90% of what we eat is digested very efficiently by our digestive enzymes. And they provide amino acids, glucose, fatty acids, vitamins, which are essential for general body health. However, there are things called residues that are resistant to digestion. They go through into the colon, and they provide the basic food for the microbiota. And as you travel down the gut, then the microbiota population explodes. As you go across into the colon, it explodes by something like a magnitude of about 7 to 8, which is an enormous number. And so we're primarily talking about a colonic situation. And there the residues undergo fermentation. And if you eat a high carbohydrate diet, then most of the residues are carbohydrates, such as fiber or resistant starch, and they undergo what's called saccharolytic fermentation. And the microbes produce butyrate, short-chain fatty acids. They also synthesize vitamins such as folic acid, iron, and B12. And they also release polyphenols from cell walls. And all of these substances are shown both in experimental models and in intact animals to have an anti-proliferative effect. So they reduce inflammation in the colon. They feed the colon as well. Butyrate is the chief energy source for the mucosa, and basically keep the lining of the colon healthy and prevent disease. On the other hand, if you have a high protein diet, high meat diet, you tend to push things towards proteolytic fermentation or putrefaction. And in that situation, you develop a different set of bugs within the colon, which produce various toxic substances such as hydrogen sulfide and ammonia-type compounds, which are actually pro-inflammatory and have been associated with genotoxicity and increased cancer risk. So that was a nice, simple way to look at things. But nowadays we're entering, the area of systems biology, which you guys are now experts at. And this is what we have to deal with. And this is an integration of all the different metabolites produced by the colon, by the microbiota, in relation to what you eat. And this just shows a pathway to try and read the different compounds there. But it's an integration of what we found in our studies, where we compare what's in the fecal samples, what's in colonic samples, and what's in the urine samples as well, all related to microbial metabolism. And you can see it's extremely complex. So there's a very tight integration of what's going on there. And it's not surprising with that going on that fermentation in the colon is going to have a major effect on not only colonic health, but body health in general. And to look at a more sort of targeted approach, we know from experimental studies and studies in humans that short-chain fatty acids, which are the product of saccharolytic fermentation, acetate, propionate, butyrate, are very good for the colon. Hydrogen in excess is toxic to all living cells, so if you can't get rid of hydrogen, then you can actually damage and produce inflammation in the colon. The way that the body does that is it has a separate group of bugs which convert hydrogen to methane and excrete it as a non-toxic product. On the other hand, if you have a high sulfate content from the high meat content, you tend to shift towards hydrogen sulfate production, and that's genotoxic. If you have a high fat diet, then as I said before, you get increased synthesis of secondary bile acids. Again, that's produced by specific deconjugating bacteria in the colon, and they're carcinogenic. The good effects are that you produce vitamin synthesis, and recent studies have actually shown that, although you produce a lot of folate in the colon, there are actually receptors within the mucosa that allow you to transport folate across and be useful food source. Polyphenols, I've spoken about it, and then toxic inflammatory substance and anti-inflammatory substances. So you can either approach this thing by looking at specific metabolites, or you can look at the whole picture. So I'm going to put most of my sort of emphasis on saccharolytic fermentation, because these are the two most important things. Butyrate production, although acetate and propionate also have been shown to have anti-inflammatory and anti-neoplastic effects. As you can see, there's this high hydrogen production, so there's a lot of gas. If you didn't have mechanisms for getting rid of that, you'd explode, and the microbes again mop that up and excrete it, either in a toxic form or non-toxic form. So butyrate is quite a remarkable metabolite. All the other cells in the body, it's, their main fuel source is glucose, but the colonocyte depends upon butyrate. And why? It obviously fills a niche because basically everything else is absorbed before it gets to the colon. You've got these residues that then liberate a nutrient, which is butyrate or short-chain fatty acids, and then why not use it for your energy uses? So that's why that situation is. But also it has these pleiotropic effects. It affects apoptosis and cell cycle regulation, principally through histone deacetylase inhibitory properties. So it actually affects transcription and cell cycling. It also affects mRNA regulation and decreases the microRNA associated with the aplasia. It also has a number of effects on reducing inflammation, mainly through its activation of GPR43 receptors and NF-kappa B downregulation. But it also affects cellular metabolism. As I said, it's the main energy source for colonocytes. And this side here is only now becoming more recognized, that it has a major role in mucosal defense. So it basically keeps the colon healthy. It restores barrier function, increases tight junction protein synthesis. It promotes mucin synthesis. Trefoil factors are involved with tissue repair. Heat shock proteins, antimicrobial peptides, again, is a very important thing. So it stabilizes the microbial communities and transglutamase activity. All of these activities, and it also has these anti-carcinogenic effects that I will talk about. So if you have a high carcinogen intake, or have a high fat intake and you produce a lot of secondary bile acids, it actually counteracts the effects of those bile acids. So there's something which is now becoming termed gene-regulating chemoprevention. And Boltman's group have been doing some very nice work on this particular aspect. And current studies and clinical trials strongly suggest that histone deacetylase inhibitors such as butyrate or trichostatinase induce expression of P21 and are effective in arresting cancer cell proliferation, lead to differentiation as in acute promyelocytic anemia or apoptosis. So it's very, very strongly associated with regulation of cell growth or epithelial growth. This is one of their latest studies where they've actually looked at notobiotic mouse model to demonstrate that dietary fiber protects against colorectal tumorigenesis in a microbiota and butyrate-dependent manner. So they did some very interesting studies where it's been shown that if you have a low butyrate intake and you produce a small amount of butyrate, all the butyrate is used for cell proliferation. So there's a sort of butyrate controversy that went on that, that if in fact, under certain circumstances, butyrate can increase growth of mucosa, but that's only if you're in a deficient diet. If you eat more starch or more carbohydrate, increase fermentation. The excess then goes into the nucleus and acts as a histone deacetylase inhibitor and actually suppresses growth. So, the quantity of butyrate that you synthesis is very important. In this study, they were actually able to show that it's the butyrate produced by the microbiota that suppressed tumorigenesis. So a lot of studies have now shown that in fact, what you eat has a major effect in changing your microbiota. In fact, if I change your diet within 2 days, your microbiota will be different. But what is the relevance? And how does that actually link to cancer health or cancer prevention? And that's the aim of the last sort of studies. Another interesting group from Australia have been looking for a number of years at the effects of resistant starch on reducing tumorigenesis in experimental models and in human subjects. And they've shown that resistant starch increases butyrate production, and it actually opposes the effects of a high meat diet on increasing proliferation. Most recently, they've done a clinical trial in human subjects where they're actually able to show that part of the mechanism of this is that butyrate suppressed production of microRNAs associated with cancer initiation and progression. So, it's all beginning to fit together rather nicely. Since we're here, I had to include that. Well, no, this is the reason that I'm here, actually, because some studies that we're doing, we're interested in integration between different dietary factors and the effect on colon cancer risk. And we're hopefully doing some studies in Alaska where they have a very high fish intake, but they don't eat fiber at all, and they have the highest colon cancer risk in the world. And so bang goes the idea that omega-3s protect against colon cancer, until I came across your study, which nicely showed that, in fact, if you add butyrate to fish oils, it actually accelerates the effect of suppressing neoplastic change. Fruit and vegetables, again, remarkably, a lot of polyphenols and micronutrients. And again, I know that I was presented some of your studies this morning showing that polyphenols and phytonutrients are so important in suppressing cancer risk experimentally. They basically are locked into cell walls, and so therefore they don't release until they get to the colon. They then are released by the effects of microbes again, and have a topical effect on the mucosa to suppress risk. So you can see that there are a number of different dietary effects. They're interacting with the microbiota to produce a milieu that actually suppresses neoplastic change. This is a study that we did where we actually flushed out the colons on healthy volunteers, and we examined the quantities of specific vitamins, which you can see at the bottom, folate, biotin, thiamine, and B12. And in yellow is the dietary intake, and in green is the quantity in the colon. You can see that in many cases, the quantity in the colon exceeds what you take in the diet, indicating net synthesis by the microbiota. And this is particularly important in Africa, where in fact there's the diet is actually not very good from a vitamin point of view, and vitamin deficiencies and folate deficiency is very common. And this, in fact, might be a natural way of getting around the problem if you give them a high fiber diet, which is what they normally take. The evidence supporting the carcinogenic role of secondary bile acids really comes from Bernstein's group, which, and this is one of their studies, showed very nicely that this is in wild-type mice, okay? And they were given secondary bile acids for 8 to 10 months, and 17 of the 18 developed colon tumors, and 10 of them cancer. And when they added an antioxidant, they actually suppressed tumor formation. So rather an elegant study showing that, although bile acids are absolutely essential for fat digestion in human subjects, an excess of fat consumption can lead to complications such as this: increased inflammation, increased inflammation, and neoplastic change. As I've said before, if you eat too much protein, and you get protein fermentation rather than carbohydrate fermentation, you can produce a lot of bioactive molecules, which are pro-carcinogens and mutagens, which can act again at a local level, at a surface level. And probably that's the most important thing, is that it's what's bathing the mucosal surface that has the predominant effect on the health of the colon. So this finally takes me to the studies that we've been doing between Africans and African Americans. And it's very interesting because I had the privilege of working as gastroenterologist both in Africa and in the USA. And working in the rural areas doing colonoscopy for things like bleeding and what have you, we might find a bleeding source, but we never ever found polyps, we never found colons, just extremely rare. And even with slight urbanization, it hasn't become a major problem yet. But as westernization increases, obviously it is going to change. Coming back to the USA, I was working in Richmond, Virginia initially with a high African-American population. And nearly every other patient that I did a screening colonoscopy on, I would find polyps, which are precancerous. So there's a striking difference between the two, as evidenced by the incidence rate differences, which I showed there. So the protocol that we did a series of studies in groups of 12 to 20 normal healthy subjects from each population, from the highest risk of that population, namely 50 to 65, which is when we all recommend that you should have colonoscopic screening. We compared differences in their diet, their usual dietary intake, colonoscopic findings, fecal and colonic microbiota, their metabolites, and we did mucosal biopsies as well to look at immunohistochemistry for CD3 and CD68 markers, and also for proliferation markers with Ki-67 as a biomarker of cancer risk. And really, you don't need to be a high-powered dietitian or scientist to see that the diets are completely different. This is a traditional African diet here. It's basically home-produced, and this is corn that is milled. It's bashed up. You saw that picture right at the beginning of my presentation. They were banging it down in bowls and pulverized, and then it's boiled. And nearly all of their food is boiled on open fires outside, and it's communist. In contrast, African Americans, in fact, all Americans are exposed to this delicious meal here, which there's no doubt that the food industry has done an incredible job in making foods delectable. And it's difficult to say no, and that's why the television promoting and so on, fast food is just so popular in the USA today. We really need to do something about that. So first of all, to talk about colonoscopy findings, this was the first group here. 9 out of 20, African Americans were characterized by polyps. 9 out of 20 had polyps. A lot of them had diverticularly. In Africans, we found no polyps at all, but we did find lymphocytic colitis and parasites in a number of subjects. And this is a segment of a tapeworm seen on colonoscopy coming out. You can see the mucosa of the colon actually is otherwise perfectly intact. And this shows a very common, surprising finding, because it almost looks like, sort of early Crohn's disease. It's inflammation, patchy inflammation, almost paptoid changes. And when we biopsied it, it basically showed a lymphocytic colitis. And there's a recent paper actually just published in American Journal of Gastroenterology suggesting that lymphocytic colitis protects against colon cancer. So, not all types of inflammation lead to cancer. Certain types of inflammation produce immunoediting and immunosurveillance, and they actually prevent against cancer. So the type of inflammation induced perhaps by parasites, and this actually shows schistosoma, which is very common in that area. You don't have tap water, you drink out of streams and things, so schistosoma, which is bilharzia, is actually very common, very prevalent. And so it's doing other things to the colon, but it's preventing cancer. But when we biopsied these subjects, and this was the first study where we compared Caucasian Americans, African Americans, and Africans, and we looked at Ki-67 staining as a proliferative marker, proliferating cells, as a recognized biomarker of cancer risk. You can see that the black dots here represent proliferating cells in both groups of Americans, whereas in Africans, they're just 1 or 2 dots, an obvious difference. And this was another subject here showing the same. This is the aggregated changes here. So clearly there was something major going on within the mucosa. And I use our justification for using Ki-67 as a biomarker of cancer risk. Basically, it's our key endpoint. Was these findings, again, in groups of normal individuals and patients with polyps and patients with cancer. And as the progression from normality to proliferation to cancer increases, so does the proliferation rate. And so, it's not the same as waiting till cancer develops, but, we've got to wait for years for that to happen. We need a marker, a biomarker, to try and give us some idea of whether you're changing the health of the colon. And when we looked at the microbiota, and this is 16S ribosomal RNA, pyrosequencing, and for those of you unfamiliar, the technique for the spectra here is that each of this is one individual. So this is African American 1, 2, 3, 4, 5, 1 to 12, the native African 1 to 12 there. And it just shows a cross section through it, and you link it all up together and get a pattern. And it gives you an idea of broad differences. And you can see that the microbiota is predominated or is dominated by Bacteroides in African Americans, whereas by Prevotella in native Africans. And not surprising, if you look at the actual species included under Prevotella, it's got a high incidence of microbes that are primarily associated with starch degradation and butyrate synthesis, such as Dialystra, Oscillospora, Succinate vibrio, and Bryolobacter. So, there are obviously major differences going on in the colon. And there was another study published in Nature where they compared microbiota of Italian children with children from the Gambia, I think it was, and it basically showed exactly the same differences. So from an early age, your microbiota are really quite different. So we published that in the American Journal of Clinical Nutrition, and what was important was that when we did a targeted approach and we looked at differences in key substrates such as butyrate and secondary bile acids, and basically we showed that in African Americans they had lower butyrate levels but higher secondary bile acids, and the reverse in Africans. So supporting our concept that maybe cancerous was being affected by the composition of their diet, because when we did the dietary analysis, as I mentioned before, the African diet had low protein, low fat, high fiber content, whereas the African American diet had the reverse. So this then prompted us to do a dietary exchange study, because it could be that there are a lot of other environmental factors that could produce this change, such as food, sanitation, housing, availability of refrigeration, and so on and so forth. So how can we be sure that this is actually diet associated? So what we did was a very unique study design where we got 20 African Americans and we put them in the CTRC for 2 weeks under lock and key and cooked for them a typical African diet and gave it to 2 weeks. And we did all of this, the studies that I've mentioned already, microbiota, mucosal measurements and so on, before and then at the end of that period. And then Africans, we did the same. We got a reasonably matched group, again of 20 subjects. We don't have CTRCs in Africa, but we found a disused lodge in the country which had cooking facilities. So we put everybody in there, including ourselves, for 2 weeks and cooked all the meals. Horrible westernized foods, barbecues every night, and delicious. They all loved it, but they're actually very keen to get back to their traditional diet at the end of it. And then we saw what happened. And the results were so spectacular that we published them recently in Nature Communications. And this is what we showed, is that the two principal changes in diet that we achieved was that native Africans were given change to a high-fat diet, 134 grams per day, and a low fiber content, 7. African Americans, on the other hand, were given the African type diet, which has low fat, 51 grams, and an awful lot of fiber, 55 grams per day. And what we looked at was our primary endpoint here, which was Chi-67 staining. And basically, when Americans changed to the African diet, staining went down significantly, and Africans to an African American diet, staining went up significantly. And this is the compound results, all 20 in both groups. Really surprising, finding. When we first did this, everybody said, oh, it's going to take much longer, 2 weeks to show any changes. People had shown that you could produce the changes in the microbiota, but they hadn't shown what effect that would have on the mucosa. And when we looked at our indices of inflammation, we looked at CD3 positive cells and epithelial lymphocytes. And again, we found this pattern that when Americans were given the African diet, the lymphocyte counts went down, and when Africans were given the westernized diet, they went up. But I mentioned already that that Africans have a high background of inflammation, and so you can see that the baseline differences were much higher in Africans and African Americans. But the direction of changes after changing the diet were in the same direction. So the high fiber diet switching down inflammation, the low fiber, high meat diet doing the reverse. And this shows the same for CD68 macrophages. I don't know what happened to this. So you have to look underneath. But basically it showed the reciprocal changes. So everything was just remarkably reciprocal. Then when we did our target analysis looking at butyrate, first of all, we looked at the functional gene for butyrate production. Must be remembered that butyrate is actually synthesized by nearly all microbiota, but there are certain groups of microbiota that are more efficient at producing than others. And so therefore, if you just look at butyrate producers with PCR, for instance, you're going to miss out on a lot. So rather look at the functional gene that is responsible for the enzyme that synthesizes the last step of butyrate. And so that's what we did here. And it showed very nicely that in African Americans, given the African diet, you up-regulated, and with Africans, there was a gross suppression on westernization of the diet. Short-chain fatty acids, basically, we showed the same differences, so that butyrate production increased when African Americans were put on the African diet, and vice versa with Africans given the African American diet. And then when we went on to look at the bad side of the story, which is bile acid synthesis, there are specific, we again looked at a targeted approach to the functional gene responsible for the production of secondary bile acids. And we were able to show the same sort of reciprocal picture. With African Americans put on the African diet, there was a gross suppression. Africans given the westernized diet, an increase, and products going exactly the same direction. So there obviously was a shift between microbes that are producing good things and bad things in both populations. We also measured, before we did colonoscopies in these guys, any of you have had the horrors of having a colonoscopy. You drink lightly and it flushes the colon out. So rather than get rid of that stuff, we collected it like true scientists. Disgusting, but it was important. And we measured the quantity of butyrate and the quantity of bile acids in them. And we found really dramatic quantitative changes associated with Africanization or westernization of the diet. With westernization of the African diet, you increase bile acid synthesis by 400%, which is really massive. So we're not talking about little subtle changes, we're talking about big differences. And I won't you know, sort of this is hard to stomach around lunchtime because there's a lot of data here, but we looked at specific bacteria that have been associated with cancer risk. And first of all, butyrate producers we've talked about. Biomarker I talked about before. Quite often with research you don't get a complete story, but in African Americans, given the African diet, there was a suppression of biomarker. Therefore, one hopes of hydrogen sulfide and inflammation risk. The same wasn't to be said for native Africans. Fusobacterium nucleatum has recently been associated with polyp formation in humans, and indeed, in native Africans, given the westernized diet, there was a progressive increase, but there was no significant change in African Americans. And these guys over here, methanogenic archaea, sulfate-reducing bacteria, and acetogens, all followed the same pattern, because what they do is that they are receptors for hydrogen production. I told you before that accumulation of hydrogen is toxic, and if you produce a high fiber or increase saccharolytic fermentation, you increase hydrogen production, and all three of these groups increased significantly, indicating that you need a true networking of microbes to actually improve the health of the colon. And looking at the microbiota in general is always very complex, difficult to follow, and suffice to say that this is the we're looking at co-occurrence network. So when one bacteria goes up, another one goes up. When another one goes down, another one goes down. So it's looking at those sort of pattern of linkages between different microbes contained within the microbiota. It gives you some idea of functional structure. And first of all, in African Americans, it's fairly tight. But after Africanization, there's a massive increase in the groups of microbes associated with butyrate production. And when we look at the other side, which are native Africans initially, again, it's a much broader spectrum, including a wider range of butyrate producers. And after westernization, you can see a collapse. So, it's really quite interesting how massive the changes in microbiota can be achieved within just a 2-week period. And when we look at the metabolome, again, this is fascinating. These are spectra by NMR. Look at this one first, as they're a little bit difficult to follow. This is native Africans, and what you're looking at, the changes, there are differences between when it's measured on the basal diet, the usual diet, and then after westernization. And you can see there's a broad spectrum, a whole lot of different metabolites produced. Highly metabolic situation when you consume a usual African diet. But you put them on the westernized diet, basically they all disappear. And the exact reverse occurs in westernized people here. First of all, not much going on. After change to a high fiber diet, an massive increase in metabolic activity. We also looked at urinary metabolites, and what was very interesting here, again, was that specific metabolites, such as these guys here, increased or were much higher in Africans at basal diet, and when African Americans were given the westernized diet, basically, there was a sudden increase in their excretion rate, and the reverse occurred when you were eating meat. So the metabolite side is very complex, as I've already said before. And when we looked at changes after Africanization or after Americanization, westernization, you can see that these are the significant changes, and they basically occurred in different directions. So, because it's difficult to see this, what they've done here is that they've grouped all the metabolites, say, related to carbohydrate metabolism here, protein metabolism over here, so it's color-coded. And so it gives you a good sense of what is actually happening metabolically in the colon after westernization of the diet. And then we obviously link the two together to see which microbes are best associated with high metabolite production. And it was noteworthy that, in fact, you can't see very well from where you are, but the X's indicate highly significant changes. And basically the association was between well-recognized high butyrate producers and butyrate levels within the metabolite. So in summary, the microbiota behaves as a community wherein intermicrobial interactions strive to produce a metabolic phenotype that supports colonic health and function. It has a genetically determined need for food residues derived from a healthy, balanced diet. The provision of an imbalanced diet leads to disturbance of structure and function, with unopposed production of metabolites that can induce inflammation and proliferation, which may increase cancer risk. Our conclusions are that we know that westernization of the diet leads to an increase of colon cancer within one generation. Our results suggest that a change in diet composition has immediate effects on the metabolic phenotype of the colonic contents associated with reciprocal changes in mucosal biomarkers of cancer risk. Our results suggest that current guidelines for fiber consumption are far too low, 35 grams per day. They should be closer to over 50 grams per day. Sounds a lot, but it's actually not. If you eat a lot of beans, you can easily get to that number. And it is basically what the traditional African diet consists of. And they also warn against progressive westernization of the African diet, where you've got this background of high inflammation, and you might in fact trigger neoplastic change by taking butyrate out of the equation. These studies can't be done by one individual, and I'm bound to my collaborators in South Africa. Nicholson's lab did all the metabolomics in London, De Vos lab in Wageningen University in Holland,